Hello po. Today I will discuss about our biochemical tests. So biochemical test is used to identify or it is used for presumptive identification of our organism. Ano po? So today's discussion sana po ay magkaroon tayo ng additional learning. Sana ay we will be able to recognize the purpose, principle, and method of each biochemical test and to be able to familiarize the different positive and negative reactions of biochemical tests. So again, biochemical test is being done to be able to distinguish the bacteria being handled to further identification to its genus and species. The further identification of bacteria is essential for the treatment of the patient's illness by giving the appropriate antibiotics. So our first biochemical test is motility test. So motility test is used to determine whether an enteric organism is motile or motile. So the principle of this is that the inoculum is stabbed into the center of a semi-solid agar dip. Bacterial motility is evident by a diffuse zone of growth extending out from the line of inoculation. Some organisms grow throughout the medium, whereas others show small areas or nodules that grow out from the line of inoculation. So the method for this is first, we will touch a straight needle to a colony of a young culture growing on agar medium. The ideal is our 18 to 24-hour culture or organism. Stab once to a depth of only 1/3 to 1 inch of the middle of the tube and incubate at 35 to 37 degrees and examine daily for 7 days. So the positive reaction or the positive result, the motile organisms will spread out the medium from the site of inoculation, while the negative result is that the non-motile or the non-motile organism will remain at the site of inoculation. But some organisms will not display sufficient growth in this medium to make an accurate determination, and additional follow-up testing is required. So, hindi lang po tayo dapat magbe-base mismo sa motility testing, but we will do or we will perform another test to identify a certain organism. So, ito po yung magiging appearance ng ating motility testing. Yung sa left po, yung ating positive reaction. Ito yung sinasabi na mag-spread out siya. Ano po? While the one in the right, ito po yung ating negative result. So sabi, non-motile organism will remain at the site of inoculation. So yung organism mo nandiyan lang po sa gitna, hindi siya gumagalaw or hindi siya motile. Ano pa? So the next one is our microdase or our modified oxidase test. It is used to differentiate gram-positive, catalase-positive cocci. So the microdase test is a rapid method for to differentiate Staphylococcus from Micrococcus species by detection the enzyme oxidase. In the presence of atmospheric oxygen, the oxidase enzyme reacts with oxidase reagent and cytochrome C to form the colored compound indophenol. Sabi, using a wooden applicator stick, Rub a small amount of several colonies of an 18 to 24-hour pure culture grown on blood agar onto a small area of microdase disc. So we will incubate it at room temp for 2 minutes, and the reagent used for this test is our 6% tetramethyl paraphenylene diamine dihydrochloride with dimethyl sulfoxide. So the positive reaction or the positive result will be the development of a blue to purple blue color or dark blue color. The positive organism for this is our Micrococcus luteus, while the negative is our Staphylococcus species. So the next is our catalase test. Catalase test is used to differentiate Staphylococci and Micrococcal species from Streptococcal species using 30% hydrogen peroxide or our so-called agua oxinada. The presence of enzyme catalase in bacteria catalyzes the liberation of oxygen and water from hydrogen peroxide. The presence of enzyme in inoculum is evidenced when a small inoculum introduced to hydrogen peroxide causes rapid elaboration of oxygen bubbles. So the positive reaction will be the production of our bubbles or effervescence. So using a small sterile wooden stick to transfer small amount of colony growth to the surface of a clean dry glass slide. So, so dapat laging malinis ang ating slide to avoid contamination. So, after that, we will place a drop of our agua oxinada to the colony. Then we will observe for the production of oxygen bubbles or effervescence. And again, the positive reaction will be gas production, bubbles production. And usually, ang ating positive organisms that causes positive catalase reaction will be our Staph aureus because ang Staph aureus po natin ay nagpo-produce ng enzyme catalase. And the negative will be our Streptococcus pyogenes. So, the lack of catalase is evident by a lack or a weak bubble production. Ano po? So, ito yung magiging itsura ng ating catalase test. Yung sa taas po natin ng ating catalase positive, makikita po natin na meron siyang copious bubbles. Ano po? While the negative catalase test, ito sa baba, mananatili siya or no bubbles produced. Ano po? So, the next is our coagulase test. Coagulase test is used to differentiate Staphylococcus aureus from coagulase negative Staphylococci. The test This test is to test the ability of certain species of Staphylococcus to coagulase plasma by the action of enzyme coagulase using rabbit plasma with EDTA. Okay, so sa coagulase test po, the preferred plasma or the preferred reagent is our rabbit plasma. Ano po? So, but in the absence of it, we can use our human plasma. So, kaya positive again ang ating Staph aureus because Staph aureus produces two forms of coagulase, the bound and free coagulase. So, ang ating bound coagulase or ang ating slide method, it is a clumping factor that is bound to the cell wall and reacts directly with fibrinogen. This results in precipitation of fibrinogen the staphylococcal cell, causing the cells to clump when a bacterial suspension is mixed with plasma. While the free coagulase or the tube coagulase, it is an extracellular protein enzyme that causes the formation of clot when S. aureus is incubated with plasma. The clotting mechanism involves activation of plasma coagulase reacting factor, which is modified or derived thrombin molecule to form coagulase CRF complex. This complex in turn reacts with fibrinogen to produce fibrin clot. So the method for our slide test, first, again, we will need a clean dry glass slide. And magdadrop po tayo, magdadrop po tayo sa magkabilang side ng glass slide ng saline and then ng ating plasma. Yung saline or yung water, we will use this as a control para makita natin kung totoong um, coagulase positive or talagang nagka-clump yung ating organism. So, with the loop, emulsify a portion of the isolated colony being tested in each drop, inoculating the water or saline first. Try to create a smooth suspension and mix well with a wooden applicator stick. So, ito po yung magiging appearance ng ating coagulase test. Makikita natin sa taas, sa right side, ito po yung ating positive test. Clearly po na may clumping factor or may clumping reaction ang ating organism dito. While sa left side is our coagulase negative organism. So, again, for the coagulase test, why is that the Staphylococcus aureus is the positive organism? Because, again, Staph aureus produces two forms of coagulase. So, we will rock gently the slide for 5 to 10 seconds. So the positive result, again, the macroscopic clumping on 10 seconds or less on coagulated plasma drop and no clumping on saline water. But pag negative po, no clumping on either drop. Mapa-plasma or mapa-saline, dapat wala siyang clumping. And then, sa tube test or sa ating free coagulase test, the test can be done or the process is being done for 1 to 4 hours. Ano po? Sa tube test, the test results can be positive at 4 hours, then revert to negative after 24 hours. Tapos, ang check natin sa ating tube test ay ang ating clot formation. So, dapat, pag positive siya sa tube test, dapat para siyang gelatin na po nagka-clot yung ating suspension. So, pag ginalaw natin, dapat hindi siya maghihiwa-hiwalay. Ano po? So, the next is our Novobiocin susceptibility testing. It is used to differentiate coag-neg Staph saprophyticus from Staphylococcus epidermidis. Because Staphylococcus saprophyticus is the only one that is resistant to Novobiocin antibiotic among these other Staphylococcus species. So, using our 5 microgram of Novobiocin and sheep blood agar, we will do our Novobiocin test. So, gagawa lang tayo ng suspension or ng inoculum of our organism, and then isi-strict natin dito sa ating agar. Tapos ilalagay natin yung ating Novobiocin disc sa gitna. So the positive result will be a zone of inhibition greater than 16 millimeter. So, as I've said, Staphylococcus saprophyticus is the only resistant Staphylococcus to our Novobiocin. So, kung makikita nyo, ito yung ating Staph saprophyticus sa ating left side. Wala siyang zone of inhibition, meaning it is resistant. While itong ating Staph aureus na meron siyang greater than 16 millimeter ay susceptible. Ano po? Sorry. Zone of inhibition with 16 millimeter. So, again, makikita nyo na ang ating Staphylococcus saprophyticus is resistant. Ano po? Wala tayong makikitang clear zone of inhibition around the disc. So, next is our Optochin susceptibility. So, Optochin susceptibility determine the effect of Optochin on an organism. So, meron siyang taxonomy. Yung Optochin natin meron taxonomy. And ano ang ating organism na merong taxonomy na protein? It is our Streptococcus pneumoniae. Ano po? So, this antibiotic inhibits the growth of susceptible organism, creating a clear or zone of inhibition around the disc. So, using an agar plate, we will streak our suspect colonies. And then, maglalagay tayo sa first quadrant ng streak ng ating Optochin disc. Bakit nga po sa first quadrant natin nilalagay? Because sa first quadrant, nagkakaroon ng heavy growth. Ano po? And then, we will incubate it for 18 to 24 hours at 35 degrees Celsius. After that, we will measure the zone of inhibition. So, resistant or positive reaction is having a less than 14 millimeter in diameter, which is our, again, Streptococcus pneumoniae. While the negative organism is our Viridans streptococci. Again, makikita natin Streptococcus pneumoniae is susceptible for having greater than 14 millimeter in diameter. Makikita natin na at ang ating strep pneumo ay mayroong clear zone of inhibition around the disc. Ano po? So, susceptible siya. And then, our S. mitis is resistant. So, wala siyang zone of inhibition around the Optochin disc. So, our Bacitracin susceptibility test is next. So, Bacitracin is used for the presumptive identification and differentiation of beta hemolytic group A strep or our Streptococcus pyogenes from other beta hemolytic streptococci. It also distinguishes Staphylococci from micrococcal species. So, bacitracin inhibits the synthesis of a bacterial cell walls. A disc, which is our taxo A, impregnated with a small amount of bacitracin, which has a 0.04 units. It is placed in an agar plate, allowing it to diffuse into the medium. Inhibits the growth of susceptible organism, and zone of inhibitions are measured after incubation. After incubation, the inoculated plates or discs are examined for zone of inhibition surrounding the disc. So, again, using our blood agar plate, kukuha tayo ng loop and we will stick 2 to 3 colonies of a pure culture. So, again, sa ating first quadrant, maglalagay tayo ng bacitracin disc using our heated forceps. And we will incubate the plate for 18 to 24 hours. And after incubation, we will also look for zone of inhibition. So, ito po yung magiging itsura ng ating bacitracin. So, meron siyang clear zone of hemolysis. Ano po? So, the positive test or the positive organism will be our Streptococcus pyogenes. Ano po? And our negative or our resistant organism for our bacitracin susceptibility test is our Streptococcus agalactiae. So, pero, etong test na to, it will depend on the integrity of the disc. So, make sure na yung ating disc or ating antibiotic is properly stored and dapat hindi pa po siya expired to be able um, to have a good result. So, dapat ang antibiotics natin ay naka-properly stored sa kung saan man siya dapat nakalagay. So, saan nga po siya dapat nakalagay? It must be on our freezer. Ano po? So, next is our CAM test. CAM test or our Christie, Atkins, and Munch-Peterson. So, itong tatlong to ang special ang ating scientists na nakadiscover na itong CAM test. So, it is used for differentiation of group B strep or our strep agalactiae from other streptococci species. So, certain organism produces a diffusible extracellular hemolytic protein or CAM factor that acts synergistically with the beta lysine of Staphylococcus aureus to cause enhanced lysis or RBCs. Group B strep are strict perpendicular to a streak of Staphylococcus aureus. So, dito sa test na to, gagamit tayo ng ating control na Staph aureus or ang ating ATCC. Ano po sabi? So, sabi, Group B strep are strict perpendicular to a streak of Staphylococcus aureus. So, ito po yung magiging itsura ng ating positive CAM test. Etong sa gitna po, ito yung ating Staph aureus. So, isi-streak natin siyang ganyan. Tapos, isi-streak natin perpendicular sa katabi nito ang ating suspected group B strep. So, kapag na-incubate natin at nagkaroon ng arrowhead appearance, ito ay positive or ito ay ang ating Streptococcus agalactiae. So, sabi, the positive result will be an enhanced hemolysis indicated by an arrowhead-shaped zone of beta hemolysis at juncture of the two organisms, while no enhancement of hemolysis is the result of our negative reaction. Again, pag may arrowhead, ito yung ating strep agalactiae. Kung makikita natin sa baba, wala siyang arrowhead appearance. ito ay negative. Ano po? But aside po sa ating Streptococcus agalactiae, may isa pa tayong organism ano po na nagpa-positive sa ating CAMP test, which is our Listeria monocytogenes. So Listeria monocytogenes and Streptococcus agalactiae tests positive for our CAMP test. So next is our V factor. V factor is used to differentiate members of Haemophilus or our fastidious gram-negative bacilli. So, members of the genus Haemophilus requires accessory growth factors of in vitro, and some species require X factor or hemin, while other requires V factor alone or both. It will only grow around the disc that provides appropriate factor for growth. So, alone of the test organism is tricked into a heart infusion broth or agar. The impregnated disc are placed directly on a confluent inoculation ano po, allowing diffusion of the accessory growth factor into the medium. So, so ito yung mga discs o requirement ng ating organisms. So, kapag meron siyang growth or positive siya sa ating X and V disc or XV discs, it might be our Haemophilus influenzae. Pero kapag meron siyang growth sa ating V disc, wala siyang growth sa ating X disc, but meron siyang light growth sa ating XV disc, it is our Haemophilus parainfluenzae. But if it has no growth or it has rather growth around the X disc and XV discs and no growth around V disc, it is our Haemophilus influenzae. So, ito po yung ating example, ang ating Haemophilus influenzae. Meron siyang, again, we will see around XV disc. So, XV disc, may growth siya. Ano po? Then, it has no growth around V disc and a slight growth between X and V discs. So, the positive result will be, or the expected, again, the growth around the XV disc only shows a requirement for both factors. But while the negative, that or the negative result, the growth over the entire surface of the agar indicates no requirement for X and V factor. So next is our bile solubility test. Bile solubility test is used to differentiate strep pneumo from alpha hemolytic strep. So kanina meron tayong Optochin test for our strep pneumo. Aside from that, we also have our bile solubility test. The principle of this is that the bile or a solution of bile rapidly lyses pneumococcal colonies. Bile salts lower the surface tension between bacterial cell membrane and the medium, thus accelerating the organism's natural autolytic process. So the method for this is after 12 to 24 hours of incubation on blood agar plate, place 1 to 2 drop of our 10% sodium desoxycholate on well-isolated colony. So, gently wash liquid over the colony without dislodging the colony from the agar. We will also Incubate the plate at 35 to 37 degrees Celsius in ambient air for 30 minutes. And after 30 minutes, we will examine for the lysis of the colony. So, the positive reaction or the positive result, the colony disintegrates. An imprint of the lice colony may remain on the zone of Streptococcus pneumoniae. Pero kapag negative naman, intact lang siya or hindi maglalays yung ating colonies. Ano po? So, ito ay ang ating Enterococcus faecalis. So, makikita nyo, yung may mga bilog po. Wala na siyang colony. Ano? Bakit? Kasi naglice na siya after dropping our 10% sodium desoxycholate. Tapos, in-incubate natin, mawawala yung colony. Pero ah, yung kanyang zone, ano siya, 'di ba, ang ating strep pneumo ay isang alpha hemolytic. So sabi 'di ba, an imprint of the lice colony may remain on the zone. So yung may mga bilog, dati siya may colony. Nag-lyse lang after lagyan ng ating reagent. Ano po? So, kapag nag-lyse yan, makikita natin na matitira na lang yung zone or yung ating pagka-alpha hemolytic. So, ibig sabihin siya ay isang Streptococcus pneumoniae. So, next is our bile esculin test. Bile esculin test is used for the presumptive identification of enterococci and organisms in the Streptococcus bovis group. The test differentiates enterococci and group D streptococci from non-group D viridans streptococci. So the gram-positive bacteria other than some strep and enterococci are inhibited by the bile salts in this medium. So organisms capable of growth in the presence of 4% bile and able to hydrolyze esculin to test esculitin. Esculitin reacts with iron and forms a dark and brown to black precipitate. So the method for this is inoculate 1 to 2 colonies from an 18 to 24-hour culture onto the surface of the slant and incubate at 35 to 37 degrees Celsius in ambient air for 48 hours. So the positive result will be the growth and blackening of the agar slant, which is our Enterococcus faecalis, while the negative will be growth and no blackening of the agar slant. So no growth or no color change our Streptococcus pneumoniae. So as a result of our nutritional requirements, some organisms may grow poorly or not all on this medium. So, ito po yung magiging itsura ng bile esculin. The positive one has a blackening of the slant. So, siya ang our Streptococcus Enterococcus faecalis. Ano po? Next is our salt tolerance test. This test is This test is used to determine the ability of an organism to grow in high concentration of salt. It is used to differentiate enterococci and non-enterococci. So, the salt tolerance test is selective and differential medium. Enterococci are resistant to high concentration of salt. So, a heart infusion broth containing 6.5 sodium chloride is used as the test medium. The broth contains a small amount of glucose and bromcresol purple as the indication of or indicator of acid production. So the pH indicator of our salt tolerance test is our bromcresol purple. So BHI or our brain heart infusion broth may be used in place of the individual components with the addition of sodium chloride and indicator dye. So, the method for this test, we will inoculate 1 or 2 colonies of an 18 to 24-hour culture into 6.5 sodium chloride broth. Incubate the tube at 35 to 37 degrees Celsius in ambient air for 48 hours, and we will check it daily for the growth. So the positive result for this test is a visible turbidity in the growth with or without color change from purple to yellow. While sa ating negative result, no turbidity or no color change. So, makikita natin sa right side ang ating positive or the appearance of the turbidity dito sa ating broth. Ano dito po sa negative, same lang siya kapag hindi pa na-inoculate yung ating colony. So, meron siyang clear suspension. Ano po. So, next is our lysine aminopeptide or ating lab test. So, this test is used for the presumptive identification of catalase negative gram-positive cocci. So, the lap disc is a rapid test for the detection of enzyme leucine aminopeptidase. Leucine beta naphthylamide impregnated discs serve as a substrate for the detection of leucine aminopeptidase. After hydrolysis of the substrate of the enzyme, the resulting beta-naphthylamine produces a red color upon addition of cinnamaldehyde reagent. So, the method for this test is, so, before tayo mag-incubate, slightly dampen the lap disc with reagent-grade water. So, using a wooden applicator stick, rub a small amount of several colonies of an 18 to 24-hour pure culture into a small area of a lap disc. Incubate at room temp for 5 minutes, and after incubation, we will add 1 drop of cinnamaldehyde. So the positive result will produce a red color within 1 minute after adding cinnamaldehyde reagent. So, pero pag negative, again, no color change. So, proceed to our methyl red or Vogue's Proscour or our MRVP. So, the combination test of MR and VP differentiates the members of Enterobacteriaceae. Methyl red determines or detects mixed acid fermentation that lowers the pH of the broth. Methyl red indicator is added after incubation. So methyl red is positive or red at pH 4.4 and yellow are negative at pH 6.2. So red is our positive result and yellow is our negative results. So, sa ating Vox Proskauer, it detects the organism's ability to convert the acid products to acetone and 2,3-butanediol. Organisms capable of using VP pathway produce a smaller amount of acid during glucose fermentation and therefore do not produce a color change when the MR indicator is added. So secondary reagent is added, alpha naphthol, followed by potassium hydroxide. A positive test is indicated by a red color complex. So organisms capable of use. So the VP detects the organism's ability to convert the acid products to acetone and 2,3-butanediol. But the MRVP testing should be used in conjunction with other confirmatory tests to differentiate organisms among the Enterobacteriaceae. So, hindi lang tayo dapat magbe-base sa ating MRVP dahil meron pang ibang test to differentiate our members of Enterobacteriaceae. So, meron tayong TSI. Ano po? So the method for this is the inoculation of MRVP broth with a drop of 24-hour BHI. So yung um plinant natin sa ating BHI after incubation, magdadrop tayo sa ating MRVP broth. Ano po? So incubate at 35 to 37 degrees for a minimum of 48 hours in ambient air. Tests should not be made with cultures incubated less than 48 hours because the end products build up to detectable levels over time. If results are equivocal at 48 hours, repeat the tests with culture incubated at 35 to 37 degrees Celsius for 4 to 5 days in ambient air. Split broth into aliquots for MR and VP. Excuse me. Excuse me po. So, but the MR test should not be read before 48 hours because some organisms, because yung ibang organisms ay hindi magpo-produce ng enough na products ano from the fermentation of glucose. So, sa ating methyl red, add 5 to 6 drops of methyl red reagent per 5 ml of broth and read reactions immediately. So, again, the positive result for our methyl red is a bright red color production. So, pag may weakly positive organism tayo, magpo-produce ito ng red-orange color. But the negative result will have a yellow color. So, sa VP naman, same lang sila ng ng color reaction. So, sa Vox Proskauer, the positive reaction will produce a red color, indicative of acetone production. But the negative result will produce yellow color. So, ito po yung ating methyl red test. So, sa positive, meron siyang red color. and the negative will produce yellow color. So, ito naman yung ating Vox Proscour. Meron siyang red sa taas. Ano po, kung makikita nyo, red. So, etong sa negative niya, wala siyang red color production. So, hindi siya hindi siya nagpo-produce ng acetone. And the methyl red, again, methyl red negative organisms may also not have sufficient time to convert products and will appear MR positive. So, meron tayong mga organisms na kahit negative, hindi siya um, or wala siyang masyadong time um, to convert the products. Ano po? So, dapat talaga ang ating reading time ng methyl red is 48 hours para may sufficient time ng ating organisms to convert such products. Ano po? Para hindi siya mag-false positive or mag-false negative. So, next is our citrate utilization test. It is used to identify organisms capable of sodium citrate as the sole carbon source and ammonium salts as the sole nitrogen source. So, this test is part of our INVIC or our Indole methyl red Vox Proscour and citrate, which is used to differentiate Enterobacteriaceae from other gram-negative rods. So, bacteria capable of growth in this medium use the citrate to convert ammonium phosphate to ammonia and ammonium hydroxide creating an alkaline pH. So, ang ating indicator or pH indicator for our citrate utilization is our bromthimol blue. So, bromthimol blue or positive will have a green color. I mean blue color. So, the principle, again, bacteria produce an enzyme, citrate permease, capable of converting citrate to pyruvate for energy production. So, bacteria capable of growth in the medium use the citrate and convert ammonium phosphate to ammonia and ammonium hydroxide, creating an alkaline pH, turning bromthimol blue indicator from green to blue. So, kahit na bromthimol blue yung pH indicator, the agar or the medium is color green and produces blue color yung positive organism po natin, ano. So again, the growth in the medium and has a blue color is our positive organism, which is the Enterobacter aerogenes, and the absence of growth is our Escherichia coli or our negative. And so, makikita natin na may production of blue color yung slant. Ano po? So, pero some organisms are capable of growth on citrate, but hindi siya nagpo-produce ng color change. So, growth is considered positive citrate utilization test even in the absence of color change. So, merong mga bacteria or merong mga organisms na tumutubo naman sa citrate test but do not produce color change. So, be careful with that organism. So, next is our urease test or our Christensen's method. So, The purpose of this test is used to determine the organism's ability to produce the enzyme urease, which hydrolyzes urea. So, Proteus species may presumptively identified by the ability to rapidly hydrolyze urea. So urea is the product of the carboxylation of amino acids. Hydrolysis of urea produces ammonium and carbon dioxide. So ammonia alkalinizes the medium and pH shift is detected by the color change of phenol red from light orange at pH 6.8 to magenta at pH 8.1. So rapid urease positive organisms magpuproduce sila ng magpuproduce sila ng pink color within 24 hours. So, ang ating pH indicator for our urease test is our phenol red. But weakly positive test or weakly positive organisms may take up to several days. Or negative result will have no color change or will have a yellow organism. So again, the positive result will have a change in color of slant from light orange to magenta for 24 hours. O pwede siyang Proteus vulgaris. So, weakly positive, it may take up to several days to become positive. So, it is our Klebsiella pneumoniae. And negative or no color change will be our Escherichia coli. So, ayan, nakikita natin na yung slant ay nagko-color pink. So, ito yung ating Proteus vulgaris. So, negative is our E. coli. So, next is our orthonitrophenyl beta-D galactopyranoside or our ONPG. So, the test of this test is used to determine the ability of an organism to produce beta-galactosidase, an enzyme that hydrolyzes the substrate ONPG to form a visible or yellow product, orthonitrophenol. The test distinguishes late lactose fermenters from non-lactose fermenters of Enterobacteriaceae. So, the ONPG requires or enters the cell of organisms that do not produce permease but are capable of hydrolyzing the ONPG to galactose and a yellow compound. O-nitrophenol indication the presence of beta-galactosidase. So, lactose fermenters must be able to transport the carbohydrate beta-galactosidase and hydrolyze B-galactosidase, the lactose to glucose and galactose. So, the method the method for this test ay gagawa tayo ng suspension. Ano po? So, we will have an aseptically suspend uh, s- organism in 0.8% of saline. Tapos, ilalagay natin sa ating sa ating tube with the ONPG disc. So, again, we will incubate it for 37 degrees Celsius for 4 hours. And then after incubation, mag-observe tayo for the color change. So the positive reaction will be the presence of beta-galactosidase or our yellow color. So it is the Shigella sonnei. Tapos pag negative naman po, colorless siya or wala siyang enzyme na beta-galactosidase. It is our Salmonella typhimurium. So, ayan yung ating ONPG positive produces yellow color. And then ang ating ONPG negative will remain colorless. Ano po, ito nasa left side. So, proceed po tayo sa ating deaminase. Phenylalanine deaminase or ating PAD test. So, PAD test is used to determine the ability of an organism to oxidatively deaminate phenylalanine to phenylpyruvic acid. The genera Morganella, Proteus, and Providentia can be differentiated from other members of Enterobacteriaceae. So, microorganisms that produce phenylalanine deaminase remove amine from phenylalanine, resulting in the presence or in the production of ammonia and phenylpyruvic acid. The phenylpyruvic acid is detected by adding a few drops of 10% colored 10% ferric chloride producing a green colored complex is formed between these two compounds. So again, it is a slant. Ano po? So, sa slant po, magsistab tayo ng 1 drop of our 24-hour brain heart infusion broth. Tapos, incubate natin for up to 24 hours. Again, at 35 to 37 degrees Celsius. So, after incubation, magdadagdag po tayo ng 4 to 5 drops of our 10% ferric chloride and observe for the production of green color because the expected result will be the development of green color on the slant after addition of ferric chloride. So, ito po ang ating Proteus mirabilis. The negative result naman, hindi siya magpo-form ng green color. So, the slant retains original color after addition of ferric chloride. So, it is our Escherichia coli. So, ito po ang ating uh, pad tests. Again, nag-create siya ng green color after the addition of our ferric chloride. So, the next is our L-pyrrolidonyl arylamidase or PYR test. So, PYR test is used for the presumptive identification of group A streptococci and enterococci by the presence of the enzyme L-pyrrolidonyl arylamidase. So, this enzyme hydrolyzes the L-pyrrolidonyl B-naphthylamide substrate to produce B-naphthylamine. So, the purpose of this test, So the positive reaction is our bright red color. It is our Enterococcus faecalis. While the negative will have a no color change or will remain orange lang po siya. Ano? Is our Escherichia coli. So kung makikita nyo, ito ang ating positive reaction. And our negative reaction will remain to its orange color. So, before reading, dapat incubate muna natin siya for up to um, 2 minutes at room temp. Ano po? So, gagamit tayo ng ating 18 to 24-hour pure culture. So, Hippurate hydrolysis is used to identify if the organism hydrolyzed benzoic acid and glycine. So, the end product of this test is the end product of hydrolysis of hippuric acid by Hippuricase include glycine and benzoic. So, the end products, glycine is deaminated by the oxidizing agent ninhydrine, which is reduced during process. And the end products of the ninhydrine oxidation react to form purple colored compound. So the positive result will be our color change. I mean, the color change with a deep purple color. So ito ang ating group B strep or our Streptococcus agalactiae. Tapos colorless naman or slightly yellow pink kapag ating Streptococcus pyogenes. So negative result ay ang ating group A. So again, the Hippurate hydrolysis test has a deep purple color. So, to proceed sa our Indole test, this test is used to identify organisms that produce the enzyme tryptophanase. So, the bacteria tryptophanase is capable of hydrolyzing tryptophan to pyruvate ammonia and indole detected by its ability to combine with certain aldehydes to form color compound. So, sa indole test, meron tayong three kinds. We have or three methods. We have the Kovacs, Ehrlich, and the spot indole test. So spot indole test is for our rapid detection. Ano? It is for the easy visualization of blue-green color compound as indole reacts with cinnamaldehyde. So this is our positive reaction. It is the production of our red color or pink to white color. Ano po? So for our oxidase test, the purpose of this test is to determine the presence of cytochrome oxidase activity in microorganisms for identifying oxygen of oxidase negative Enterobacteriaceae, differentiating them from other gram-negative bacilli. So the principle of this is to determine the presence of bacterial cytochrome oxidase using the oxidation of substrate tetramethyl p phenyldiamine dihydrochloride to indophenol, a dark purple colored end product. So, no color development indicates a negative test and absence of enzyme. So, the positive will produce a dark purple color within 10 seconds, and negative will remain colorless. So, ayan. So, ating oxidase test, makikita nyo na pag nilagay yung organism sa ating oxidase disk, magkakaroon siya ng purple reaction or purple color. So, most of our members of Enterobacteriaceae are oxidase positive. Ano po? So, sa ating triple sugar iron naman, again, it is used for differentiation of the members of Enterobacteriaceae from other gram-negative rods. So, the principle of this is to determine whether an organism is glucose fermenter. Ano po? So, ang indicator or pH indicator nito ay phenol red na nagde-detect ng ating acid production. While the H2S production indicator is our ferrous ammonium sulfate. So, anaerobic decomposition through glucose utilization wherein the entire bath turns acidic and slant becomes acidic when lactose and sucrose are being used up. So, ito yung ating mga K over K, K over A. So, sa ating, pag sinabing K over K, meron siyang alkaline bath and alkaline slant. While our K over A, meron siyang alkaline slant and acid bath. Tapos, sa A over A, meron siyang acid slant and acid bath, which means na yung organism ay nagfe-ferment ng glucose, sucrose, and lactose. Etong glucose, sucrose, and lactose, siya yung mga carbohydrates na ginagamit natin or na meron ng ating triple sugar iron. Ano pa? So, ang TSI, meron siyang 10 parts of lactose, 10 parts of sucrose, and 1 part of glucose and peptone. So, ang black precipitate in the bath indicates the production of ferrous sulfide or itong H2S natin. So, kapag may black precipitate siya, meaning it is H2S positive. Ano po? So, ayan. Gas formation, meron siyang bubbles or cracks sa gitna ng ating agar. Meron siyang blackening of the agar again kapag H2S positive. So, all members of Enterobacteriaceae are enterogenic or gas producers except Shigella. Ano po? So, sabi, ang Pseudomonas, E. coli, Kleb, all are gas producers except Shigella. Magkaiba po yung gas production and H2S production. Ano po? So, ang H2S positive naman po natin, ito ang ating space. Ano? Yung Sriracha, Arizona, Providencia, Citrobacter, and Edwardshella. Ano po? Pero yung gas production, um, napoproduce siya ng ating Enterobacteriaceae, sorry, except for our Shigella. Ano po? So, ito yung ating iba-ibang appearance ng ating TSI. So, etong may blackening dito sa may right part, ito yung ating mga H2S production or H2S positive organisms. So, meron siyang red slant, meron siyang yellow butt na hindi na masyado nakikita. Meron siyang gas or crack. And then, meron siyang H2S production. So, di ba sabi dito kapag um, may kapag alkaline, it is yellow, and kapag acid, baliktad. Kapag alkaline, it is red color, and kapag acid, it is yellow color. So, eto sabi red slant, which means it has a um, alkaline slant. Ano po? So, itong sa gitna naman po, nakikita natin may crack siya sa gitna, meaning it produces gas production. Meron siyang gas production. So, yellow siya meaning uh, A over A or acid over acid. Wala siyang H2S or blackening of the agar. So, dito naman sa may left side, ito yung ating uh, K over K. Ano po? Because it has a red slant and red butt, wala siyang crack or wala siyang gas production, wala rin siyang blackening, meaning wala siyang H2S production. So, yan po for our TSI. So, for our LIA naman or our lysine iron agar, the purpose is to determine the ability of an organism to determine lysine decarboxylate lysine and produce hydrogen sulfide. So, principle is the lysine iron agar contains lysine, peptones, and small amount of glucose, ferric ammonium citrate, and sodium thiosulfate. So, it is also useful for the identification of our Enterobacteriaceae. So, if the decarboxylase is not produced, the bath remains acidic or yellow. But if the amination does not occur, the Lia slant remains purple. So kung ang K sa ating TSI ay color red, ang K naman sa ating Lia ay color purple. And ang ating A ay ang ating yellow color. So pH indicator for our phenyl for our Lia is our bromcresol purple. While sa TSI, ang pH indicator po natin sa TSI is ang ating phenyl red. Again, the H2S indicator is our ferric ammonium citrate. So, kapag ang slant natin ang nagde-detect if the organism is capable of lysine deamination. So, kapag red siya or positive, it is our ppm, Proteus, Providencia, and Marganella. While pag negative or K is our purple color. So, kung ang slant ang nagde-detect ng lysine deamination, ang BAT naman ang nagde-detect ng lysine decarboxylation. Baliktad naman sila, ang purple naman ang ating positive reaction, while the yellow is our negative reaction. So, as a result, kapag K over K, meron siyang lysine decarboxylation, wala siyang glucose fermentation. Ito ang ating Escherichia coli. Pero kapag naman, meron siyang K over K and H2S positive, meaning meron siyang lysine decarboxylation, wala siyang lysine deamination, and wala rin siyang glucose fermentation. Ito ang ating Salmonella, Arizona, Citrobacter, and Edwardsiella. So, pag may K over A naman, wala siyang lysine decarboxylation, wala rin siyang lysine deamination, and meron siyang glucose fermentation. Ito ang ating Shigella. So, pag R over A, wala siyang lysine decarboxylation, wala rin siyang lysine deamination, but meron siyang glucose fermentation. Ito ulit ang ating ppm. Ito ang ating Proteus, Providencia, and Marganella. So, for the reactions, ito ang ating K. Sa right side, ito ang ating K over A or plus H2S. Meron siyang blackening again of the agar or the medium. So, dito sa K over K, purple lang po siya because nga meron siyang, again, lysine decarboxylation. So, ito ang ating E. coli yung sa left side. So, that's all for our biochemical test. Sana po ay may natutunan tayo. Thank you po.